Welcome, everybody. It's 10:02 Pacific time. I think we have almost 60 people joining our Zoom meeting today. This is a joint BAGIM and SAGIM event to hear from Dr. Julian Michel. I would like to start by thanking all of our sponsors who help make BAGIM and SAGIM possible. Without them, we would not be able to have any of these talks. Our sponsors include OpenEye, with Cadence Molecular Sciences, CCDC, XTAL-Pi, Schrödinger, Zifo, Disk Engine, and in particular, I would really like to thank Crescit for sponsoring today's talk. Today, Julian Michel will be talking about allostery under the lens of molecular dynamics simulations. Julian Michel is a professor of biomolecular simulation at the University of Edinburgh and director of the Open Biosim Community Interest Company. He has over 20 years of experience in academic research in molecular simulation methodologies and their applications to simulations of biomolecules. He has a long-standing track record of collaborative research with the pharmaceutical industry and is a member of the academic consortia CCP Biosim and HEC Biosim. I'll let Julian take it away. I believe that he is open to questions and lively discussion during his talk. So feel free to unmute and ask questions or add them in the chat, and I'll try and keep an eye on that. Welcome, everybody. Thanks for joining us today.
Okay, thanks, David. Yes, so looking forward to lively discussion, but keep an eye on the clock. Right, let me see if I can share my screen so we can get going. And how is the screen looking for you? You're happy for me to...
Quite completely full screen, but I can see it fine if anyone can't.
Okay, yeah, I mean, with my setup, I think if we keep it like that, that's probably easier for me.
Okay.
So unless someone has issues, let's go ahead.
Okay.
Okay, cool. All right, David. So yeah, thanks for the invite, the opportunity to talk a bit about some of the research ongoing in my group at the University of Edinburgh. So today's talk, the theme, I guess, is allostery. It's mainly two-story, and then you will see that there is obviously allostery as a common thread. So before we jump into matters, let me just tell you a little bit about the kind of focus that my group has currently at Edinburgh. So I've been an academic at Edinburgh for about 12 years, and over that period, I guess our research interests have evolved somewhat, although they've always have at heart biomolecular simulation methods and how we can develop methods that can find application for drug discovery. And today, I guess our current research interests mainly fall under three areas. So conformational dynamics and function, so how dynamics of biomolecule is linked to their function. That's a long-standing topic in biomolecular simulation, of course. And then increasingly, we're exploring opportunities for advance in machine learning to change the way we carry out our traditional biomolecular simulation. And then as the technology has evolved, so to speak, and nowadays you would say there's a lot more practitioner and user of molecular dynamic simulation, we increasingly are looking at how we can take scientific science in free energy perturbation and convert that into well-engineered solution that can be used more broadly. All right, but today's talk will largely focus on the first area of conformational dynamics and function. So there's two things that are recurrent throughout this talk. And the first is protein allostery. And so now I'm pretty sure that most of the people in the audience are very familiar with the topic here, but just to be very clear, what are we talking about here? We're talking about studying the behavior of molecules that bind at so-called allosteric sites. So these are binding sites at the surface of a protein that are somewhat distant, remotely from the actual active site of, depending what you look at. So this is a kinase. Many of you have probably recognized that. And so kinase would need to bind ATP and a substrate, and so the catalysis occurs somewhere around here. So here we've got an example of a compound that binds somewhere towards the back of the kinase and somehow inhibits the reaction, right? And so the question is, how does that work? Can we explain it? Can we anticipate this behavior? Because if we can, then we open up more opportunities for modulating protein function with molecules. And the kind of main techniques that we use to investigate this problem is molecular dynamics simulations. So why? Well, because molecular dynamics simulation give us a unique opportunity, I would say, to characterize in full detail allosteric mechanisms. It gives us an opportunity to catalog conformation that biomolecule can adopt and then relate structure to function, so that here we have a picture of an energy landscape of this kinase adopting different conformation in the presence of different inhibitor that may be associated with different activities. And nowadays we can really recast the problem of understanding allostery as understanding perturbation of protein energy landscape. And what we want to get out of this kind of techniques is a picture that give us insight about structure, but also thermodynamics, kinetics, and the link to function. So with that, let me start with the first story here, which was published a few years ago, which is probably it's a well-studied system where allostery manifests through point mutation in side chain that change packing behavior. And that appears seemingly simple, but there's some interesting complexity that we can hopefully shed light on. So what we're looking at here is an enzyme called Cyclophilin A. This is a kind of cartoon representation of the Cyclophilin A protein. You would have the action, the catalysis happening somewhere around here, the active site region. And what does this enzyme do? Well, they isomerize amide group in proline residues, so they can accelerate conversion between a trans and cis form of proline. So there's a whole bunch of different cyclophilin isoforms with a very interesting biology, and they've been around for some time, but there's still some open question about their behavior and their role in disease. But cyclophilin A has also emerged over decades as a good model system for biophysical study, because it's shall we say, reasonably easy to work with in a biochemistry lab. So now the matter I want to focus on is this data that was published a few years ago by Otton et al., which looks at the catalytic efficiency of cyclophilin A and different variants that feature different mutation. And what you see here is, so this is a plot of Kcat over Km, so loosely you can take it as a measure of catalytic efficiency, so the higher the better. And we're looking at the different peptide substrate that can get processed by this enzyme. So where we obviously undergo turnover and isomerization around the amide group of the proline. So what you see is so wild type, that's what it does, fairly efficiently for different kind of model substrate. But if you introduce, and if you look more closely at the active site, you can see a model of the substrate AAPF. So how can we explain catalysis in this enzyme? Well, there's some key residues that tighten interaction around the transition state, and there's some electrostatic stabilization afforded by arginine 55, and you see the amide group in front of the proline residue here. So you kind of see in purple side chain that are in contact with the substrate, and a little bit behind in green, so that's kind of second hydration chain or second layer. You have this sidechain in green here. So if you watch out serine 99, for instance, here, you can mutate it into threonine, and if you do that, it actually has quite a large effect on the catalytic efficiency of cyclophilin A. And then if you go further and taking this serine to threonine mutant, and you introduce additional mutations, cysteine to serine, and here isoleucine to valine, you can recover somewhat the catalytic efficiency that was present in wild type. So the question is how does a mutation here propagate to affect catalysis effectively? Now, what has been shown is that if you carry out NMR relaxation dispersion experiment on cyclophilin A and this mutant variant, you can resolve motional process that involve a large cluster of residue. And basically what the experiment tells you is that there's a whole bunch of residue, about 1/2 of cyclophilin A, that are undergoing collective motion on a millisecond time scale. And so that patch of residue include several of the side chains that I've just highlighted on the previous slide. And interestingly, that there is so out of this millisecond motion, you can extract from the NMR experiment things like rate constant for conformational change between so-called major or minor conformation. So it doesn't tell you exactly what they look like. It tells you there must be an equilibrium between two states, and there's some conformational change happening on the order of a second time scale. And it appears there's a relationship between the rate of transition between the two states and the catalytic efficiency. So then that suggests that the millisecond motion are involved in the catalytic cycle somehow. Now you get a little bit more structural insight through room temperature X-ray crystallography experiment, which are able to resolve different rotameric state for some of the key side chain involved in the substrate binding. So phenyl 113 in particular packs against the proline residue, and in the wild type cyclophilin A at room temperature, you can clearly resolve two distinct rotameric states that vary in their chi-1 angle. So we can call the in blue one the in state, and in brown the out state, because of the relative motion of the phenyl ring. And this motion or these experiments are done on APO form, but it's close enough to that you could expect there's going to be some fallback effect on motion position of arginine 55, which we know is implicated in catalysis. Now, if you solve the structure of S1990, what is striking here is that you only observe the out rotamer here. And so that gives another clue, suggesting that, yes, you could have perhaps out to in motion that have been observed here, and that explain the loss of catalytic activity in S1990. Now, initially, we were actually studying cyclophilin A for unrelated reasons, but because we were working on this system, we were familiar with the literature I've just gone through. And what we observed in some of our MD simulation is that actually we didn't really have to work really hard to observe transition of F113 between the different chi-1 rotamer. So this is a simple movie from a typical MD simulation of a few hundred nanoseconds at room temperature, which clearly shows several transitions between these different chi-1 states. And so that piqued a bit our curiosity, because there was this literature suggesting this transition occurs on a second or so, or hundreds of millisecond time scale. So clearly not something we would expect to capture routinely in our simulation. But yet it was happening here. So we decided to investigate a bit further the problem. And to do that, we took a more systematic approach where we decided to build Markov State Models of cyclophilin A side chain dynamics in the active site. So we could not just get anecdotal evidence of transition between state, but have a better handle on thermodynamics and kinetics. And so for that, we had to carry out simulation of the dynamics of apoform of wild type, and then this 3 mutant variant. So briefly, Markov State Model, I won't really go too much into detail, but the idea here is that we can capture the long time scale dynamics behavior of a system by carrying out ensemble of independent MD simulation that are each on the relatively short time scale. But we can cluster data from the different simulation, typically done in parallel, along a subspace that can resolve different conformation of interest. And then through some form of clustering schemes, we can coarse grain the data into a smaller set of states. And the rule here is that the clustering has to be done such that these are typical snapshots from MD trajectory, and you want basically every point here within one cluster to interconvert between each other much more rapidly than they transition between other cluster. And then if you can do that, then you can actually recover equilibrium distribution and long time scale kinetics properties. Now there's of course a lot of caveat, but that's the main picture. So this is kind of data that we get out of this MSM analysis, where here we've taken the final model and just looked at a very simple view of what is the equilibrium likelihood of observing phenyl 113 in the out or in rotameric state. And so you see in blue here, this is result from the MD simulation, which shows there's about same likelihood of observing the out in state. What you see here in a dashed dotted line is the occupancy that we observe for this rotameric state in crystallographic structure at room temperature. So you've got dark blue here and light blue here. So this appears for wild type fairly consistent with room temperature X-ray. I mean, you see 2 rotamer in the X-ray, and there's clearly 2 rotamer that have similar probability in the MD simulation. Then moving on to the S99 threonine mutant. So here we've done this point mutation. What we observe is that because of the increased bulk of threonine, we don't observe, we barely observe the in rotameric state. We only observe out state. And that's consistent also with the crystallographic data that only resolve 1 rotamer. Yeah? And then if you go down, there's data for all the mutant, but if you look, say, at the triple mutant, STCS-IV, what we observe is a picture that's also fairly consistent with crystallography. In the case of the triple mutant, there's a small probability of observing the hint state that you can also observe in the occupancy data with a similar ratio. So what that shows us is that we seem to capture quite well the density that you observe in the crystallography in this simulation. Now we can go a little bit further with the MSM approach and also extract kinetics about the rate of conversion between the in and out state of phenyl 113. And you've got a kind of summary data here of the equilibrium probabilities and rate of transition for out to in and in to out. And now the striking thing about this result is that you can take KAKB, extreme exchange rate, and everything on the order of hundreds of per microsecond. So that's further evidence that we see this flip happening very rapidly, and we can explain the X-ray data, but we cannot really relate that to the NMR millisecond timescale motion, because our model predicts that these flips are happening about a million times faster than what NMR suggests. So then we decided to carry out and ask ourselves, okay, so if we take the different state that we observe in our simulation, can we characterize the catalytic activity of cyclophilin A in these different substates? And we've done that using fairly established simulation techniques. So we're using umbrella sampling here to derive rotation of the omega angle so that we can force our model peptide substrate to go from trans to cis states, and then we can carry out simulation at different values, and then using WAM construct the free energy profile for the isomerization reaction. And it happens to be one of very rare enzyme where you can reasonably satisfactorily study this process using a classical force field, so that makes our life easier. So what do we learn from this PMF calculation? So if we start with the final 113 in state, so you see in black, that would be the uncatalyzed profile. So there would be a preference for proline to sit in a trans state, and there's only a small amount of cis that form, and a barrier on the order of 20 kcal or so. So that's consistent with experiment, and largely because we're using a force field that's been parameterized to get this right. And now if you look at the effect of the enzyme on this reaction, you can see that there is catalysis that accelerates trans to cis conversion. It occurs via the omega plus 90 degree transition state. There's no difference between wild type and S1990 in this calculation. So as long as F113 is in, it doesn't matter whether you've got serine or threonine right behind. Next, if you look at the profile with phenyl 113 out, then you see there's no catalysis that you can observe compared to the uncatalyzed reaction in solution. And the key here is, again, we can't see any difference between wild type and ST in this plot. Now we can explain why F113 out is catalytically incompetent. If you look at simulation data around the transition state region, and say in the case of F113 in, you can see there's a network of hydrogen bond in interaction that really keeps the transition state substrate in place. These are the hydrogen bonding probability for these different donor-acceptor pair. And most of these are completely disrupted if you force Chi1 rotamer to flip to F113 out. And that that's relatively easy to explain, because if you put F113 out, it actually pushes against the proline ring of the substrate. And therefore it sits a bit further in the active site, and as a consequence, all the hydrogen bonding interaction are disrupted, and you have no longer preferential stabilization of the transition state. So if we put all of this together, what we have is a model where we have in wild type cyclophilin A rapid equilibrium between inactive F113 out state and F113 in state. And only essentially only once the enzyme adopts this conformation, it can catalyze the isomerization reaction. And this motion happens on the transition on the order of microseconds. In the case of the mutant form, what we see is that the equilibrium favors the inactive state. We still see transition, still on the order of microsecond. It's a bit slower, but we are nowhere near the millisecond regime. And if we plug now the data from the activation free energies we get from the umbrella sampling profile and the MSM data for the equilibrium probability, we can actually calculate the ratio of bidirectional isomerization rate between wild type and the mutant. And the reason we do that is that this can also be done from the actual experimental data. And what that shows is that our model predicts decrease in isomerization rates. That's consistent with the measurements, although we've got larger uncertainty, largely due to the imprecision of our activation free energies. So if we summarize, it seems like there's two conflicting proposals for allosteric effect of point mutation in cyclophilin A. What we see in our MD simulation is a fast motion of F113 and a fairly local conformational change, whereas NMR has observed slow motion on millisecond time scale that involve large number of residue. What we can say is that this motion in our end are sufficient to explain the change of catalysis. So how do we really resolve this forward? Well, that's a good question. One thing we can actually do is make some prediction based on our model that potentially could be followed up by experiment. So one of the main features of our model is that we see rotameric flip on the time scale of hundreds of nanoseconds to microseconds. And this sorts of timescale formational process is, in principle, is accessible to NMR. It's not the easiest one to work with because you can resolve fast dynamics using kind of relaxation experiment on a kind of timescales that are under the tumbling time of protein. And then much slower using relaxation dispersion, you can look at process on the millisecond time scale. And kind of in between window, in principle, could be experiment that involve residual dipolar coupling measurements. That could be done, we argue, on Chi-1 label sidechain that should generate a signature. This is signature predicted in change of S2 order parameter calculated with different averaging window. So today, that's a prediction that's tend to be tested. But hopefully we'll be able to resolve this question later on. Okay, so with that, I'll move on to the second story of the talk. And so this is much more recent work that we're in the process of publishing. Where this time we're interested in assessing the allosteric effect of ligand rather than mutations on enzymes. And the idea is that we still want to use the same framework of molecular dynamics simulation and Markov State Model, but we would like really to have a general framework that's applicable to different classes of enzymes and conformational change happening on a large range of length and time scale. So the idea is that we would like a tool where imagine that you have done a simulation of an enzyme bound to a substrate pre-catalysis, and you're able to generate ensemble of snapshots and then classify them according to whether the enzyme is in a catalytically active or inactive state. And then you see an example here where most of the time we've got an active state, and then about 25% of the time we've got an inactive state because in that case this is a phosphatase, and for the cleavage of the phosphate group to happen, you need this loop to close and come in close proximity to the phosphate group. So if we were able then to throw ligand anywhere on the surface of the enzyme and repeat this calculation, we could check, have we changed the likelihood of observing an active or inactive state? This is a case where it hasn't. So we would say this compound might bind here, but it's not going to influence the mechanism. Whereas perhaps another somewhere, if it decreases the active state probability, we would say this compound would behave as an inhibitor. So, so to develop this framework, we've been studying in depth the enzyme PTP1B. So that's phosphatase. That's a kind of model enzyme for studying phosphatase. So PTP1B, in order to cleave phosphate group from phosphorylated tyrosine, you see the small substrate peptide here, needs a so-called WPD loop here to go from open to a closed conformation. Now this loop motion happen on time scale of several microseconds, and so here there's no doubt that this doesn't happen very quickly in MD simulation. So we know we're gonna need a slightly different approach to capture that. There's also good experimental data about the effect of mutation on allostery. And there's a data set of known compounds that bind in different places and that are known to allosterically inhibit the reaction. So you've got here a pocket between these two helices that's occupied here by either compound 1, which is a potent allosteric inhibitor. Compound 2 is a fragment that's obtained by basically chopping off the aryl sulfonamide group here. And that's an extremely weak allosteric inhibitor, the limit of what you can detect in the assay. But you can also allosterically modulate function through covalently tethered fragment that bind around here. And then to throw into the mix, we've also looked at a compound that have been observed by high-throughput X-ray crystallography experiment to bind to the surface of PTP1B, but where it hasn't been possible to characterize whether this compound would have an allosteric effect. And it's typically because you're looking at low molecular weight fragment, weak binder. You're not really able to assess in a functional assay this compound without beefing up potency a little bit. So in the case of cyclophilin A, we were fortunate that the kind of motion that were of interest to us were happening spontaneously, so over the time scale of equilibrium MD simulation at a relatively affordable cost. Here a bit less so. So we have to actively push the WPD loop if we want to observe spontaneous loop, if we want to observe loop motion that go from active to inactive state. And so the workflow we put together involves first running steered MD simulation, where you go from, say, active loop conformation to inactive loop conformation and vice versa. And save along that steered MD run state that then are used as an input for equilibrium MD simulation, such that we kind of bias our sampling so that we can resolve the motional process of interest and then throw the data sets into the MSM machinery to obtain information about population and kinetics. So how does that work in a slightly more detail? So you generate trajectory data for your system. It could be in the presence of different compounds and so on. The trajectory data is featurized so that we can have a low dimensionality representation that we need in order to build statistics for building our MSM. And so we choose collective variables that can resolve position of the WPD loop. We're also looking at, for instance, things like distance between a phosphorylated tyrosine substrate and the catalytic cysteine. Then we do a global clustering of all the data that we obtain, so unliganded, liganded, sim variant, and so on. And we do that so we have consistent cluster center, so to facilitate comparison between different simulations. So there's some details here in the protocols that are important to minimize variability between simulations. And then out of that, we get things like probabilities, equilibrium probability of observing the system at this different cluster center. And that gets further coarse-grained into a simple two-state definition of active or inactive. And from there, we can translate summing the equilibrium probabilities of all these points. We can give a simple answer like this enzyme is active 27% of the time or something like that. Now, I should stress, we are now looking at much more complicated motions, and there's a lot of noise in the data set that we generate. And so to have a handle on that, we do bootstrap resampling of the whole procedure, which allow us to build probability distribution of this figure, and that help us having an indication of how precise the results are. So we put together on a GitHub repo the whole pipeline by putting together several different biomolecular simulation tools and then changing them So for those of you who are interested, you can take a look at Michel Lab AMO repository. Now, before I discuss some of the results, I want to stress that this is not straightforward work. And there was a lot of back and forth that was needed in our hand before we obtained satisfactory results. So the kind of most naive thing you would do if you start a project, which is what we did to begin with, is you take PDB structure of the active-inactive form of PTP1B, and then you set up a steering run where you steer according to, say, a backbone atom RMSD of the WPD loop. Now, if you do that, that's not actually a very robust way of simulating a conformational change, because there's some details that are important to correctly go from an inactive to active state and vice versa. So there's some other side chain that have to rearrange as you close the loop. This is not necessarily obvious at first. In that case, we know this because there is prior mutagenesis data that can guide us. But when you apply this to a less characterized system, that's I will be the first to say that it could be a challenge. There's also a challenge for us is that as the WPD loop goes from inactive to active state, it is known experimentally that in PTP1B there's a C-terminal alpha helix that is disordered in the inactive state and that folds and form a helical state in the active closed state. And it's quite challenging for us to reversibly simulate disorder order transition of the helix as we drive a closure of the WPD loop. And so here, in order to obtain consistent and reproducible steering run between active to inactive and vice versa, we made the decision to actually simulate a truncated variant that has removed helix alpha 7. And I'll come back to that in one slide or two. Now, you can see some example here of MSM results where we have been quite careful. So, this is maybe version 3 or 4 of our protocol where we steer according to the loop, but also the allosteric network. And if we do that, compared to, say, this is the active state distribution for PTP1B bound to substrate, and then repeated with additionally a compound that's supposed to behave as an inhibitor, we see a downward shift in active state probability. So that would make us happy. And that's reasonably reproducible between completely different repeat from scratch. And if you're not really careful about the steering protocol, where you only steer the WPD loop, what happens is you have hysteresis between inactive to active and active to inactive transition. And we completely lose the effect of observing destabilization of the active state. So that shows how we have to be careful about how we set up this biased simulation. So with this caveat aside, these are the kind of the main result from the study. So you have here, these are every time different setup of PTP1B. So this is a simulation of PTP1B without any substrate or inhibitor. And then we see a low likelihood of closing the WPD loop, which is fairly consistent with experimental data. It suggests it's only a few % of the time that the WPD loop closes if there's no peptide substrate. If you have a substrate peptide, we have an increase in active state probability, so from about 5 to 25%. That's a partial result in the sense that experimentally we would expect the WPD loop to be mostly closed if you model if you have the substrate present. But if you recall, we're also working with a variant where we've truncated helix alpha 7 because we couldn't sample it reproducibly. And that variant experimentally has been shown to only partially activate WPD, the PTP1B. And so overall, we take that as a fairly good result that we're able to reproduce the shift in preference for the inactive state conformation when the substrate is bound. Now, moving on from there, all the other simulations have the substrate peptide modeled in and some other compound present somewhere else on the surface of PTP1B. Now, if we start with compound 1, you can see we've got a downward shift in active state probability. It's a fairly tight result. So that's clear behavior that this compound behaves as an allosteric inhibitor. Same goes for compound 3. That's our covalently tethered fragment. And then we've got some other compounds that don't really show strong behavior as an inhibitor. So if you have a look at compound 2 first, compound 2 is the fragment that you obtain by chopping off half of one effectively. Now, what we observed here in this simulation, when we looked at what was happening, actually having chopped off half of compound 1, compound 2 is such a weak binder in our simulation that it spontaneously unbinds in many of the simulation we run. And so that models a bit the interpretation of this plot. So we repeated this calculation using loose positional restraint to enforce compound 2 to remain in the allosteric pocket. But having done that, we still don't really see much of allosteric effect. So we can zoom in and try to gain some insight about this. So you can see here compound 1, when it's bound between these two helices, you can see it has this kind of U shape where it's wrapping around phenylalanine 280. And then compound 2, because we've basically removed this part, cannot wrap around phenylalanine 280. So we were looking for insight, structural insight. Why in our simulation this is predicted to be an inhibitor and is not? And one thing we spotted is that the behavior of F280 is quite distinct in the simulation. So in the case of PTP1B bound to 1, F280 mainly adopts this rotameric state where it's pointing upwards here because it's packed against the ligand. But in the case of 2, it's quite loose. It can basically flap around a fair bit. Now that's not a difference that you can spot from crystallographic data where the side chain in public data, at least for PTP1B, has been resolved with the same Chi1 value. But now if you look also at the behavior of PTP1B without inhibitor, we can still separate the conformational ensemble into closed open loop states. And we can see that the correlation between being in the inactive state or the active state and a shift in relative probability of the Chi1 angle value for F280. Now, I will be the first to say that doesn't mean that we can fully explain the allosteric mechanism, but it's giving us a clear evidence of difference in the behavior in the active site that we can correlate to different functional outcome. And so that could suggest some application. For instance, if one wanted to do mutagenesis experiment, this would be an interesting residue. Or if one wanted to do virtual screen using structure sample from the simulation, this could give you a way to select structure that may help you enrich selection of inhibitor. We've also looked at the other pocket that was binding covalently tethered fragment. And so you can see, so experimentally, this is a weaker inhibitor than compound 1, but it does show some measurable effect. And that's qualitatively reproduced in this plot. What we did as an experiment is to break the covalent tether and simulate a non-covalent variant of this fragment. So that we could assess, does the tether itself has an effect on the allosteric property of the fragment? And what you see is that when we remove the tether, we actually have much weaker inhibition, or it's almost back to baseline. And if we look at the behavior in the simulation, we see that the fragment actually no longer adopt the binding mode observed in the case of the covalently bound variant, but it bends sideways and offer completely different orientation and interaction with the side chains along the helix. And so that suggests that one has to be careful if you take a covalently bound fragment and then try to recover a non-covalent variant, because the tether itself may actually have driven the functional outcome. And finally, you see this is our fragment binder observed by crystallography with uncharacterized functional effect to date. You can see in our simulation, it's quite noisy behavior, and we also need weak positional restraint to keep it in place, but it doesn't really suggest potential for strong inhibition. And if we look at how these fragments interact, we see that it engages PTP1B in a manner that's consistent with the major pose of the untethered fragment that has shown a decreased potency. So we would take this result together to suggest that it would be better to optimize compound, for instance, using this binding mode to begin with, and engage this side of the pocket to retain and improve the allosteric effect. Okay, and so with that, really I've shared two stories of MD simulation and protein allostery, and how we can hopefully learn about how to harness protein dynamics to create new opportunities for ligand design.
Thank you very much. I haven't seen anything in the chat, so I'll just open it up to questions now.
Okay.
On my part, I'll say that the theme of your talk, the title of your talk was Allostery, but I thought a nice theme was also just the phenylalanine rotamers. It showed up in both
Yes.
the two stories, which was really nice. And it's not something that I think that much about. And I also don't think that much about loop conformations. Usually when I'm looking at the protein structures and I see loops, I don't think that that's something that's going to be moving on the order of microseconds or slower milliseconds. You know, I usually think that there's a very low energy barriers for loops to reorient. And so I found that part particularly illuminating. And I thought that it was very interesting that you showed that the loop, that's creating the simulation to get the loop to sample both conformations requires careful setup and tuning of the simulation. And that if you're not careful and you truncate in the wrong place, you don't get the right dynamics. I wouldn't have expected that naively, and I thought that was a really nice and clear result. I thought it was interesting, the probability distributions that you got as well for those different modulators. I would not have thought of looking at how they bias the protein conformation just by binding in that way, binding between, you know, and changing the equilibrium distribution between states. So I thought that was really nice. But I see that in the chat, someone has raised their hand. So rather than me ramble about all the stuff that I thought was really nice and informative, I'll let Zane ask their question. Zane, I cannot hear you. I'm not sure. I don't think you're muted, but I could not hear you. I could just see your mouth moving. If you'd like to type it in the chat, I'd be happy, or someone else can read it out loud.
While Zane sorts it out, can I ask a question?
Sure, yeah, go ahead.
Yeah, thank you for your talk. I was, really intrigued by your mentioning when you did the MSM analysis in part 2 of the talk, you had many different ligands, and you mentioned how there's this step where you had to carefully combine sort of the state spaces of the different groups of simulations? Could you speak a little more on that, please?
Yes, so it may not be strictly essential, but what you have to be a bit careful when you compare MSM built for different variants of the same system, because if you cluster independently each set of trajectory, you'll have different cluster center, and that creates a little bit of noise. And that will also, when you coarsen the data to have simple two-state description, you'll end up with different boundaries between the two states. Now, if you have well-behaved case where say you have one microstate with a clear lump of density, another microstate with a clear other lump of density, it probably doesn't create a big variability. But when the data is a bit noisy, which is often the case when you do this kind of large-scale loop motion, or you don't have very, very long data set. When the data is noisy, we see that there is more variability in the clustering and the impact of clustering on the state probabilities. So to ease comparison here, their approach was to do a global clustering of all the data so that you have a common set of cluster center that then can be used for the comparison. So that helped tighten up a little bit the probability distribution. Yeah. Right. Hopefully that answers your question.
Yes, thank you.
I can read out Zane's question that he typed in the chat. If it is, can you speak to using MSMs that require longer timescale simulations?
So longer than microseconds, say, for instance. I think it's the same. So I think there's two approaches really. So either you can take kind of brute force approach, or there's also adaptive sampling protocol that will try to intelligently generate batches of trajectory so that you can ultimately hopefully span large conformational change. We have in other work I haven't talked about, we have used MSM and some kind of biased MD scheme to access this kind of longer time scale. I would say that I think it's always possible as long as you can drive a conformational change. In principle, you can connect state that would be connected by very long time scale. One issue you have is that the model may be incomplete. It may have overlooked. There's no reason that say the only thing happening to WPD on a multi-microsecond time scale is only the WPD loop moving. Now I touched on disorder order transition we neglected. And so what that means is that you have to be careful, I think, how you interpret the output of the model. So the absolute state probability may be misleading, although the ratio between the two states that you obtain should be consistent. And the timescale for the transitions themselves, I would expect them to be a bit faster than experiment if there's other process that haven't been resolved by the model. So probably in an absolute sense, it might be difficult to connect with experiment, but as in the case study we looked at here, what we needed is a way to compare different variants of the protein and how that affects the same motional process. So in that case, I think that's safer ground.
I think we have time for one more question, if anyone would like to ask Julian.
Yeah, I can chip in here. Julian, thanks for a great talk on obviously a very complex problem. I'm naturally thinking about practical utility of such a classification system, and I wonder if you could speak to like the kind of throughput available for this kind of intense treatment of
Sure, yeah.
screening molecules.
So I would say actually generating the MSM is reasonably quick and parallelizable once you're happy with the steered MD protocol. And so that part is difficult at the moment in terms of rolling this out quickly, because you need to have time to become familiar with the system that you're simulating, to pick on details. So that is difficult to document, because I would say, and we're in the process, we're studying several types of proteins with the same framework, not only enzyme. We're also looking at large-scale domain motion, or we're looking at membrane proteins, where every time the motional process that govern allostery are not easily accessible by a kind of routine MD regime, and so they require this biased MD protocol, which will depend a lot on the degree of expertise of the modeler and the time available to set things right. But if you're happy with the steering protocol, let's say, or some other form of biased MD to connect the state of interest, then you can run that fairly rapidly and repeat that with many different ligands.
I think there's one more question in chat I'll read out. How did you pick your collective variables for steered MD from SIND?
Yeah, so a little bit of trial and error, as I explained in the story. Starting with a simple idea. Okay, let's drive the RMSD of the backbone atom in the WPD loop, and then realizing that's a problem because we observe inconsistency in the conformation sample if you do it independently from active to inactive due to some of the couple sidechain rearrangement that need to happen. And if we are basically closing the loop too fast, not allowing the sidechain to rearrange, and then you haven't reached the correct active state, but some form of high energy variant. So trial and error and then knowledge about the system, I think is the answer. And then you can always come up with some schedule that steers things in a certain sequence, so that you start with what everybody agrees is, we call that inactive, and then you end up with something that looks like the active state according to experimental data.
Okay, great. Thank you so much. We are at the top of the hour, so I will let you get back to your evening. Thanks everyone for joining us. We'll see you next time.
Okay, right. Thank you, everyone. Bye.